Hello everyone, this is Dr. Pooja, your biochemistry mentor. In today's video, we are going to discuss the metabolism of sulfur-containing amino acids. So, let's start with the discussion. Metabolism of sulfur-containing amino acids. Now, what are the sulfur-containing amino acids? Obviously, if we talk about the definition, the amino acids which have sulfur in them will come under this category. So, there are two amino acids which belong to this category, cysteine and methionine. Right. Now, what is the structural difference between cysteine and methionine? Both are having sulfur in them. And again, we will come to the main structure of the 20 standard amino acids. As I have been mentioning in my previous videos also, this is the generalized structure. This is your alpha carbon atom, which is connected to sulfhydryl group in cysteine. And if we talk about methionine, methionine has again same structure like this, but the alpha carbon here is connected with CH2, CH2, SCH3. So this becomes the structure of methionine. And if we talk about this, it has a thio-ether linkage. And if we talk about this, this is a thio one, or this is a thioalcohol. It is also known as a sulfhydryl group. Right, very important. And if you see, the difference is, if we talk about this, this is CH2SH and this becomes CH2CH2SCH3. That means methionine has a CH3 attached to S, or sulfhydryl group in methionine, it is not free. Okay, so this is the basic difference. And just because of this difference, if we have to categorize the properties, cysteine is non-essential, methionine is essential. Right, and due to this sulfhydryl group, cysteine comes under polar, but obviously uncharged. And this comes under non-polar. Then if we talk about the metabolism as per carbon skeleton, this cysteine is also glucogenic and methionine is also a glucogenic amino acid. So I would say both come under the category of glucogenic amino acids, right? So these are the three main important features. Now the question is, if I am saying methionine as an essential amino acid, so what role it has to play as an essential amino acid? So methionine is required for the synthesis of cysteine, or I can say that cysteine is derived from methionine. Cysteine is derived from and the most important characteristic feature of methionine is that methionine is having a methyl group, as you can see here, and this methyl group serves as the transfer of this methyl group, and it is required for the synthesis of so many important compounds just by catalyzing a transmethylation reaction. This is a very important feature of methionine. So, second important point is methionine is required for transmethylation. required for transmethylation. Very important, and always remember, whenever a transfer of methyl group takes place, it is an exothermic process. So transmethylation is a exothermic process. So these are the three main important points about cysteine and methionine. Now we all know that whenever any amino acid, whenever anything has to work, it has to be converted into its active form. Same is the case with methionine. Methionine, whenever it has to transfer this methyl group, it has to be converted into its active form. So first of all, we are going to take up the metabolism of methionine, how methionine is required for transmethylation, and how cysteine is derived from methionine. So first of all, again our concentration would be on the anabolic reactions, then on the catabolic reactions, and then on the inborn errors of metabolism associated with methionine. So first of all, let's take methionine. Now see, the active form of methionine is SAM. So the active form is SAM, which stands for S-adenosyl methionine. Very important. Now the question is how methionine is converted into SAM. So the name of the enzyme here is MAT. So first of all, I'll just draw that cycle. So what happens is, this methionine is converted into SAM first, which is S-adenosyl methionine in the presence of MAT. MAT is the name of the enzyme, okay, and adenosyl will come here. Now once this SAM donates the methyl group, because its purpose is to donate the methyl group, so methyl group will be accepted by the acceptor, and this S-adenosyl methionine will be converted into S-adenosyl homocysteine. Function is over, whatever we wanted we got it. Then S-adenosyl homocysteine will release this adenosyl group and it will be converted into homocysteine. And this homocysteine will be converted into methionine later. So this entire cycle is known as regeneration of methionine, because methionine is simply giving the methyl group and it is converted back to methionine. Okay, so if we talk about the enzymes here, you can see MAT is the enzyme here, and this MAT stands for methionine adenosyl transferase. Methionine adenosyl transferase. And this methionine is, MAT is of three main types: MAT1, MAT2 and MAT3. Liver, and 2 is present in the extrahepatic tissues. Very important, right? So this is the enzyme which is required to convert methionine into its active form, and if this enzyme is absent, it always leads to a boiled cabbage odor in urine. The urine will give the smell of boiled cabbage. Okay, so this is reaction 1 when methionine is converted into SAM. Now this SAM, once it gives the methyl group, it is converted into S-adenosyl homocysteine, as this is what I have written, and this will be converted into homocysteine. Clear? And methionine is regenerated back. Here the enzyme is adenosyl transferase. Now the question is how homocysteine is converted into methionine and how cysteine is derived. Okay, so homocysteine to methionine requires B9 and B12, which we are going to discuss in detail now. And coming over to this homocysteine, this in the presence of B6 will form cysteine. So you can see that just by seeing this particular cascade of reactions, this methionine is forming, donating the methyl group, and itself is converted back to methionine, right? So this is actually known as a regeneration of methionine. So first of all, if we talk about the example of transmethylation, so what are first of all the examples of transmethylation? Transmethylation. So many reactions we have also covered. Just if you remember the reaction, conversion of norepinephrine to epinephrine, right? Second is conversion of guanidoacetate to creatine. This is again a very important reaction, right? Then conversion of ethanol amine to choline. These are the reactions which we have studied, which we have covered. That's why I am telling you these are the most important reactions which are actually taking place in the presence of methionine. And sometimes we say that methionine is that amino acid which gives rise to 1-carbon metabolism. 1-carbon metabolism is transfer of this methyl group, right? So this is again very important. Now we are going to study that how this methionine is produced from homocysteine and how this homocysteine is being converted to cysteine, because cysteine is a non-essential amino acid which is synthesized from methionine, which is an essential amino acid. And in this entire episode, homocysteine also plays a very, very important role, because nowadays homocysteine is considered to be a marker of MI also. Even though estimation of homocysteine requires, it's like standards are very costly, so it requires so much of money, but yes, it is an indicator of MI. Okay. So now, first of all, how this homocysteine is converted into methionine. First reaction is this. And if you see the reaction clearly, I have mentioned the role of 2 vitamins here. And what are those vitamins? B9 and B12. Okay, and always remember one thing, that B9 and B12 are the only 2 hematopoietic vitamins which are present. Okay, and this is one reaction in which B9 and B12 are actually acting as best friends with respect to one another. Okay, now see, if methionine is giving a methyl group, it is being converted into homocysteine. Clear example is this: if we put methyl group into homocysteine, it will be converted back to methionine. As simple as that. Now the question is from where this methyl group comes. Okay, so always remember B9, which is folate. It is a donor of methyl group. So I would say methyl donor is B9 and B12, which is cobalamin, it is simply functioning as coenzyme just to transport this, and the enzyme is methionine synthase. No ATP required, simple reaction, methionine synthase. Okay, so this is how homocysteine will be converted into methionine. Now, if we see the reaction, I can simply say that this is homocysteine and this is methionine, right? And B12, methyl B12, when it donates its methyl group, it will be converted into B12. And from where this methyl group is coming? It is coming from methyl THF, which will be converted into THF like this. So this is how the reaction is going on, where you can see that methyl THF is giving the methyl group to B12, and this B12 will give the methyl group to homocysteine so that it can be converted into methionine, right? So if we talk about the enzymes here, I can simply say that methionine synthase is the enzyme here, and here if we talk about, it is methyl tetrahydrofolate reductase, right? Or commonly we can call it as MTFR. And here 𝗖𝗵_सा, it is methyl cobalamin. Transferase, or you can say methionine, because this is just acting as a coenzyme, right? So this is how homocysteine is converted into methionine. Then we come over to the second reaction. I told you there are three vitamins which are taking place in the entire process. So the second one is how this homocysteine is converted into cysteine. Homocysteine to cysteine. Now, if we talk about this, we can simply say that this homocysteine, this is a very important reaction, it combines with serine. Serine, which itself is a non-essential amino acid. So this serine will come in the presence of B6. We all know B6 is PLP, which is the most important coenzyme in fact 95% of the reactions related to the amino acid metabolism. And the principal reaction, transamination, which is actually the fate of all the 20 standard amino acids, I mean except 3, they require B6 as a coenzyme. So B6 is the coenzyme, and the enzyme here is cystathionine beta synthase. So homocysteine, when it combines with serine in the presence of cystathionine beta synthase, it is going to form cystathionine. And this cystathionine splits up. This splits up in the presence of cystathioninase or cystathionine hydrolase to give cysteine, which we require, and it gives homoserine. This homoserine can later be converted to form succinyl coenzyme A. And cysteine also can later be converted into, you can say, glucogenic amino acid. It forms pyruvate in the end, right? So we can say that homocysteine, while being converted into cysteine, requires B6 as the coenzyme, right? And if we talk about this reaction, we can simply say that methyl B12 and methyl THF, and these are the enzymes. So this is how the regeneration of methionine continues, first thing, right? And this is how cysteine is synthesized. Then there is one more molecule which is cystine. So what happens is that this cysteine is converted into cystine. And what happens here is two molecules of cysteine, because pronunciation mein there is a difference, and you will see there are so many similes in biochemistry also. So two molecules of cysteine forms one molecule of cystine. And if we talk about the enzyme here, enzyme is cysteine reductase. And what happens here is it is going to form a disulfide bond, right? So this is how cysteine is converted to cystine. So if I have to give the summary of the entire metabolism of methionine, I can simply say that this methionine forms homocysteine, right, which is converted back to methionine, homocysteine which can form cysteine, and cysteine which can form cystine. And methionine is responsible for the conversion of this. And for transmethylation, if we talk about, then just now I told you epinephrine is produced, choline is produced, so many other compounds are produced with the help of methionine, and the most important one is creatine. Creatine, which is the energy reservoir of the muscles, this is also produced, right? So this is about the metabolism of methionine. Then after this, then we come over to the inborn errors of metabolism associated with that. So what are the inborn errors of metabolism? Right, now we are going to take all the steps one by one, because here you have seen that there are 3 vitamins which are playing a major role. Now coming over to the first reaction, if we talk about the first reaction in which homocysteine is forming cysteine. Sorry, homocysteine, one second. Pehle we will take how homocysteine is being converted, then later on we will take how homocysteine is being converted into cysteine. So first reaction is homocysteine is forming methionine. Right, here we saw the role of 2 vitamins which I told you, that CH3, B12, and second is methyl THF converted to THF, like this. Okay, now the question is, and the name of the enzyme is methionine synthase. Suppose if I say either this will be absent or this will be absent, right? So whether B9 is absent, whether B12 is absent, it will lead to increased concentration of homocysteine and decreased concentration of methionine. It's clear from the picture itself. So I can say that if B9 or B12 is deficient, it will lead to homocystinuria, means increased concentration of homocysteine. So I can say that it will lead to homocystinuria. Now what happens in this homocystinuria? If we see it like this, as per the diagram, I can say that there is increased concentration of homocysteine and decreased level of methionine. But there is no effect on cysteine. Very, very important. Cysteine mein no effect will be there. Okay. So we can say that whether B9 is absent, whether B12 is absent, it will lead to homocystinuria, and this type of homocystinuria is known as non-classic homocystinuria. Right. Now if we look at this reaction, there are again two aspects which will come. Pehle I'll just explain it. Now see, two vitamins are playing a major role in this, or they are both the hematopoietic vitamins. So now if we say in general, whether B9 is absent, whether B12 is absent, it will always give rise to increased concentration of homocysteine and decreased concentration of methionine. But how we are going to differentiate between a patient who is suffering from non-classic homocystinuria, and we need to know that whether it is due to B9 deficiency or whether it is due to B12 deficiency, because both are linked together. If we see, since both are hematopoietic vitamins, so I can say megaloblastic anemia will be seen in both the cases, but how to differentiate, right? Now the question is, how to differentiate a patient which is B9 deficient suffering from non-classic homocystinuria, B12 deficient who is suffering from non-classic homocystinuria. Now see here, suppose if I say B12 is deficient and B9 is normal. There can be two scenarios. If B12 is deficient and B9 is normal, now what will happen to that patient? Folic acid ka concentration is normal, so folic acid will keep on donating its methyl group. Now the question is, B12 is deficient. That means the one who has to accept that folic acid, or who has to accept that methyl group, is absent. So here it will lead to folate trap. This is very important. Folate trap simply signifies that there will be entrapment of folic acid, means folic acid will not be free to give its methyl group to other reactions. Getting my point? Because what is happening, B9 doesn't know B12 is deficient. So B9 will come and keep on giving its methyl group. So there will be an increase in the methyl group, but the acceptor which has to accept that methyl group is absent. So it will lead to trapment of folic acid, right? So I can simply say that since B12 is deficient, B9 will give its methyl group, right? So other reactions requiring B9 will suffer, right, which leads to folate trap. And if I say ki B12 is deficient, one more reaction here is that the patient will suffer from methylmalonic aciduria. Why? Because methylmalonate always requires B12 for its function. So I can say that methyl melonic aciduria will be present. Is present. This is also one of the very important features. And if we talk about why methyl melonic aciduria, because again, if we talk about this reaction, this propanol coenzyme A, we all know that this propanol coenzyme A, which comes from degradation of breakdown of odd chain fatty acids, in the presence of carboxylase, forms methyl melonyl CoA. And this methyl melonyl CoA forms L-methyl melonyl CoA. Right, and this L-methyl melonyl CoA forms succinyl coenzyme A. And this reaction requires B12 and the enzyme is mutase. So if B12 is absent, it will lead to increased concentration of methyl melonic acid in urine and the patient will suffer from methyl melonic aciduria. Okay. So what happens is, if B12 is deficient, B9 is normal, there will be trapment of folic acid, there will be trapment of methyl group and the patient will suffer from methyl melonic aciduria. This is very, very important. And if we talk about the another scenario, if I say that B12 is present, B9 is absent, if this is the case, again homocystinuria will be there, same. Okay, but the only thing here will be how to differentiate. Here methyl malonate levels will be normal. So this is the basic difference between B12 and B9, and obviously both the patients will have megaloblastic anemia, because again you can say DNA synthesis will be the one which will be affected. Right. So this is your non-classic homocystinuria. Right, which is due to the deficiency of B12 or I would say B9. Then second category of a reaction is in which homocysteine is being converted into cysteine. Homocysteine to cysteine. Now, when we look at this reaction, we have seen that this homocysteine combines with the serine, okay, and B6 is the enzyme, coenzyme, and cystathionine beta synthase is the enzyme, and what it forms? It forms cystathionine. And it will be converted back to homoserine and cysteine. Right. Now what happens is, if I say this B6 or cystathionine beta synthase is absent, again what will happen here? Here there will be increased concentration of homocysteine, clear? But there will be decreased concentration of cysteine. And if we talk about the level of methionine, methionine will be normal. Why methionine will be normal? Because homocysteine will be converted into methionine. There is no hindrance in that. So I can say homocysteine can be converted into methionine. So methionine will be normal. And this is the basic difference between B9, B12 deficiency and B6 deficiency. So again, if we talk about this, it will lead to, so I can say that if B6 is deficient, I can say that there will be decrease deficient, or you can say this enzyme is absent, cystathionine beta synthase. There will be increased concentration of homocysteine leading to homocystinuria, and if we talk about the levels of cysteine and methionine, it will lead to increased homocysteine, decreased cysteine, and normal methionine levels. Right, and this is known as classic homocystinuria. So I can say that classic homocystinuria is due to the deficiency of B6, and non-classic homocystinuria is due to the deficiency of B9 or B12. This is very, very important. Right. So if we talk about the biochemical investigation, what are the first of all tests? Okay. So if we talk about the biochemical investigations, we can say that cyanide nitroprusside test will be positive. Cyanide nitroprusside test will be positive in both. Second is tandem mass spectrophotometry will be present. With the help of this we can see. Now how to differentiate between these three types of homocystinuria? So for that I can simply say that if we talk about the enzymes here, right, three features here, homocystinuria obviously here, urea. Here classic, right, non-classic. One more enzyme is there which is methyl tetrahydrofolate reductase. So if we talk about क्लासिक में डेफिशिएंसी किसकी है? B6 डेफिशिएंट. नॉन क्लासिक में B9 और B12 deficient, and here reductase enzyme is deficient. Okay, so if we talk about cysteine levels or methionine level, cysteine level in classic will be low, but methionine level will be normal. If we talk about non-classic में cysteine level will be normal and methionine level will be decreased. And regarding reductase में it won't make any difference to cysteine or methionine level, but yes, methionine level we can say it might be reduced. Okay, so this is one very important feature biochemically, how we can distinguish between classic and non-classic as per the concentration of cysteine and methionine. And if we talk about B9 and B12, always remember in B9 deficiency, methyl malonate levels normal, will be normal, and coming over to this, the most important again, megaloblastic anemia will be there. Anemia present. And if we talk about B12 deficiency, and if we talk about B12, I can simply say that methyl malonate levels abnormal, or methyl malonate levels will increase, and megaloblastic anemia will be present. Anemia present. So this is how we have to differentiate, and the third important point here will be folate trap. So this is the major difference between B9 deficiency and B12 deficiency. Then methyl malonate levels normal in B9, and here methyl malonate levels increases and it leads to folate trap. Folate trap, understanding the concept of folate trap is very important, because if B12 is deficient, B9 will keep on donating its methyl group and rest of the reactions are going to suffer. And this is a very important one. Okay. Now then we come over to what is homocystinuria and what are the clinical features in homocystinuria. So what happens here is, initially the patient is totally asymptomatic. When the person is born, asymptomatic. Is asymptomatic, right? But at the onset of say 3 years, at the age of 3 years, the deterioration starts. Now what happens is, there is a developmental delay, right? So what happens here is, the vision starts reducing. So there is a reduction in vision. Later stages mein it leads to ectopia lentis. What happens in this ectopia lentis? The position of the lens dislocated medially and downwards. Very important, right? Then skeletal deformities are there. Skeletal deformities are there, like arachnodactyly is there. Right, and the person, he becomes conscious. So what happens is, his fingers and toes, everything is like increasing in the length. Arm to length ratio also increases. So what happens, he becomes conscious and he bend down his, that spinal cord also, leading to scoliosis. So what happens is the person starts becoming conscious कि he is abnormally growing so much tall, and his fingers are moving beyond that. So this all leads to a kind of skeletal deformity, and he starts bending and starts walking. So it resembles Marfan syndrome also, and it is actually known as Marfanoid habitus. Marfanoid habitus. Very important. Means it resembles Marfan's syndrome. Resembles Marfan's syndrome. And another feature is, since there is increase in the concentration of homocysteine, I told you homocysteine is considered to be a marker of MI also, so it leads to thromboembolism. Right, and I'll just show you the picture of a person suffering from this. Right, you can see that, just check arm to height ratio, it is comparatively long, but he is looking totally like a abnormal person, okay. And you can see scoliosis, because he has that kind of, he becomes conscious and his posture changes. And if you see the toes, if you see the fingers, and joint hypermobility is there. Then breastbone dips inward or protrudes outward, you can see that also. So, totally this is known as marfanoid habitus. Very, very important, which is a characteristic feature of only and only homocystinuria. Clear? Whether it is classic, whether it is non-classic, but I have differentiated between classic and non-classic what happens. And just now I told you कि if it is non-classic, so non-classic में B9 and B12 deficiency is there. So whenever a patient comes, so obviously one has to see all these factors before making any diagnosis. Right, because again we cannot play with the life of the patient. So whenever a patient comes, we have to go for physical examination, we have to go for the biochemical investigations, blood reports, so many things are there before giving the thing. Up coming over to this, if we talk about the treatment, for example, if we talk about classic patient suffering from classic or a patient suffering from non-classic homocystinuria. So simple is treatment in classic homocystinuria, or in general we will discuss कि पूरे में क्या है. So if we talk about this, in homocystinuria, if it is classic, we know it is B6 deficiency, so we can put that patient on B6 supplementation. So B6 supplementation will be there. So that at least cysteine can be produced, because if cysteine is not produced, cystine will not be produced and concentration of homocysteine will increase. And then another treatment is, if we talk about non-classic, non-classic में it is either due to B9 or B12 deficiency. So depending upon this, whatever is absent, supplementation of B9 or B12 is given. Right, and second important here is betaine supplementation. What is betaine? It is trimethylglycine. Betaine supplementation. Right, so betaine is trimethylglycine, so basically it will act as a remethylation of methionine. So betaine ka purpose is that betaine remethylates methionine. How it is remethylating methionine? Simply homocysteine will be converted into methionine by providing methyl, which is coming from betaine. And rest is vitamin supplementation. So this is homocystinuria, classic or non-classic. Then we come over to another important disorder which is related to the sulfur containing amino acid metabolism, which is cystinuria. Now in this cystinuria, basically what is the reason behind the cystinuria? Here there is defective tubular reabsorption of the basic amino acids. Defective या defect in tubular reabsorption of the basic amino acids. Right. So if there is a defect in the reabsorption of these amino acids, automatically they will not be reabsorbed and they will be excreted in urine. And it is actually known as CoLA. So you can say that CoLA, CoLA, Coca-Cola. So CoLA mein C stands for cystine, not cysteine. I am writing it because most of the students get confused between this. Cystine, O for ornithine, L for lysine and A for arginine. Right. So I can say that CoLA, these amino acids will be excreted in urine. So this is cystinuria. Okay. Defect in the tubular reabsorption of the basic amino acids cysteine, ornithine, lysine and arginine. And this cystinuria is a part of Ghayrod's tetrad. Very, very important again. Part of Ghayrod's tetrad. And who is this Ghayrod? Ghayrod was basically you can say a scientist or a doctor, 𝗧𝗲𝗵𝗺𝗺, who gave the concept of tetrad, which is again the mnemonic is CAP here. Very, very important, where 𝗲𝗵𝗵𝗺. C stands for cystinuria. Again cystinuria, CoLA. Another A stands for 𝗵𝗵𝗵𝗺, which we have studied in detail. The deficient enzyme is homogentisate oxidase. Then another A is albinism, where the defect is in tyrosinase. And P stands for pentosouria, where we can say the deficient enzyme is dehydrogenase, xylitol dehydrogenase. Right. So we can say that cystinuria, which is accompanied by CoLA, which becomes a part of Ghayrod's tetrad, which is CAP, where C stands for cystinuria, A stands for 𝗵𝗵𝗺𝗺, A stands for albinism and P stands for pentosouria. So this is the importance of the sulfur containing amino acids, right. So if we have to just give a brief gist of this, I can simply say that this methionine, which is responsible for the transfer of this methyl group, helps in the synthesis of epinephrine. The most important one is creatine, very, very important, okay, and choline, and it is easily converted to homocysteine and converted back. And this homocysteine is converted into cysteine, which is converted into cystine. And if we talk about the inborn errors of metabolism, the most important ones are homocystinuria and cystinuria. One more is there, Oosthuis syndrome we call it, Oosthuis syndrome, which is actually due to the defect in again the transport of this methionine. So, and if we talk about this, methionine is a glucogenic amino acid. Similarly, cysteine is also a glucogenic amino acid. And if we talk about the test to differentiate between this, the test is lead acetate test, right? Lead acetate test given by only cysteine, not methionine. Because methionine ka structure, if you see, methionine has SCH3. So this bond cannot be broken, right? So we can consider only cysteine by, we can estimate cysteine only by this lead acetate test. So this is all about the metabolism of sulfur-containing amino acids, cysteine and methionine, where methionine is a strictly non-polar amino acid. It is essential. Why? Because it is required for the synthesis of cysteine, which is a polar but uncharged amino acid. And this cysteine helps in the formation of cystine. And don't get confused between cysteine and cystine, because the spelling only makes a word of difference. Right? Thank you so much.